So we'll see what is the rationale for drug targeting. Having understood the problems encountered when we take the dosage form orally or parenterally, right? We have to concentrate on how this targeting will help us to achieve the required concentration of the drug. So the site-specific target drug delivery negotiate an exclusive delivery to the specific pre-identified compartment with maximum intrinsic activity of the drug and concomitantly reduces the access of the drug to the irrelevant non-target cells. Okay. Then the control rate and mode of the drug delivery to the pharmacological receptors and specific binding with the target cells, as well as bioenvironmental protection of the drug en route to the site of action are specific features of the drug targeting. So you are getting the drug delivery direct to the target and you are protecting the drug till it reaches to the target. So this is the important thing in the case of the drugs which gets degraded till they reach to the target. So you get higher concentration of the drug at the site of the action and resultant lower concentration of the drug at the non-target tissue where the toxicity might crop up. Okay. There is a relative cellular uptake of the drug vehicle, then the liberation of the drug, efflux of the free drug from the target site, it, it is taking place. Okay. Then the targeting is signified if the target compartment is distinguished from the other compartments where the toxicity may occur and also if the active drug could be placed predominantly in the proximity of the target site. So this way you can increase the concentration of the drug at that particular target site. Then the restricted distribution of the parent drug to the non-target site with effective accessibility to the target site could maximize the benefits of the targeted drug delivery system. So here we'll see how this simple drug and the targeted drug delivery system is acting. So this is a free drug and you can see this is a solid line and this is a dotted line. So when you got the free drug, okay, it goes to all the sites, all non-target sites, okay. So they are able to access this non-target sites. They have got affinity to for this non-target site. What is the effect of this? Effect of this is a toxicity. So this dotted line indicates that it is not going to the target site only, and because it has not got a very great affinity or very great access to this target cell. So therefore, just how it is going to the other cells, that is non-target site, same concentration it is going to even to that particular area where it is required to exert its effect. And therefore, you don't get a specific effect, you get a very limited effect. Other thing is that what are the bioenvironmental factors? Okay, like for example, acidity in the stomach, the microflora, the enzymes. So these bioenvironmental factors, they also leads to inactivation or it will leads to less therapeutic effect. So these are the limitations which are associated with the free drug. Now, in the case of a drug carrier system, so your drug is present in a carrier system. So this carrier system doesn't get affected by the bioenvironmental factors. Why? Because of the sequestration and improved therapeutic availability. For example, you just said that enteric coating. So because of the enteric coating, it is getting uh, protected. So from the bioenvironmental factors, so there is a sequestering of the drug and therefore there is a improved therapeutic availability. After it reaches the blood, after its even these protection from the hepatic metabolism also, what will happen is now it will go to the target site. Specifically, it will go to the target site. Why? Because of the facilitated transport. Okay. So that because of that, you will get proper targeting there. And this dotted line indicates that it is not going to the non-target site. It is not going to the non-target site because there is no access, there is no specific affinity. Why? Because the affinity to the target site is more, access to the target site is more. So therefore, the amount of the drug going to the non-target area is very less, and therefore you will have very limited side effects. So this is how we compare the simple free drug and the drug which is present in the carrier. So therefore, we need to know what is this drug carrier. It is very important component of this targeted drug delivery system. So carrier is one of the most important entities which is essentially required for successful transport of the loaded drugs. These are the drug vectors which sequester, transport, and retain the drug en route while elute or deliver it within or in the vicinity of the target. So these vectors or the carrier should carry the drug without getting it degraded, and it should transport the drug to that particular target, and after it reaches the target it should slowly release the drug near that particular target. So these are the require this is the importance of the drug carrier. So what are the features of an ideal drug carrier? So the drug carrier should be able to cross the anatomical barriers, and in the case of tumor chemotherapy it should cross the tumor vasculature. Then it should reach to the target site, no, so it should get recognized by that target. So therefore it should be recognized specifically and selectively by the target cell, and and it must maintain the avidity and specificity of the surface ligand. That is, it should be going to that particular specific organ, and it should give you the required action there. It should give the required action there. So avidity and specificity of the surface ligand is very important. Then the linkage of the drug and the directing unit, that is the ligand, it should be stable in the plasma, interstitial, and other biofluids. Like for example, you have taken the drug delivery system and you have attached some ligand, that is also called as a homing device. So this system, you plan that it should go to this particular target site, but what is the medium through which it is going to the target site? For example, if you're administering the formulation by the parenteral route, it is going in the blood, so in the plasma it should be stable. From the blood, by extravasation, it comes out of the blood vessels and it goes in the interstitial fluid. So in the interstitial components also it should be stable, and if it is going to some other biofluids, for example in the cerebrospinal fluid, so it should be stable in that biofluid as well. Next, it should be non-toxic. The carrier itself should not cause any toxicity reaction. It should be non-immunogenic. It should not cause any immunity reaction, and it should be biodegradable. It should get degraded in the system. It should be biodegradable particulate or macromolecule, and after recognition and internalization, the carrier system should release the drug moiety inside the target organ, tissue, or cells. So it should go specifically to that particular target. For that it should get recognized, and after that it should release the drug in that particular target area. Now, the biomodules which are used for carrier navigation and site recognition should not be ubiquitous, otherwise it may cross over the sites, defeating the concept of targeting. It should not be ubiquitous means it should not be general, means it is going everywhere in the body. It should not happen that it is going everywhere in the body. It should be specific. It should go to that particular site only. So these are the features of an ideal drug carrier. Now we'll see what are the various carrier systems used for targeted drug delivery. First, colloidal carriers, like for example, vesicular systems like liposomes, neosomes, virosomes, immunoliposomes. These are the vesicular systems. Then microparticulate systems. Okay, this can be your microspheres or nanoparticles, right? So you know vesicular systems, they have got a particular shape and the dimensions which is not there in the case of particulate system. This can be of any geometry present. Okay, then Cellular carriers like resil erythrocytes, serum albumin, antibodies, platelets, leukocytes. So here you can have these as a carriers for directing the drug. Then supramolecular delivery like micelles, reverse micelles, liquid crystals, lipoproteins. Okay, very low density lipoproteins, low density lipoproteins. These can be used. Then polymer based delivery systems like mucoadhesives, biodegradable, bioerodible, soluble synthetic carriers, they can also be used. Then macromolecular carriers like proteins, glycoproteins, neoglycoproteins, monoclonal antibodies, polysaccharides, these also can be used as a carrier.